Welcome to Vegetation Remote Sensing, a video series developed as part of the Advanced Remote Sensing and GIS course at the Australian National University. I am Marta Yebra, and in this video series, I will present some examples of applications of remote sensing to assess vegetation. But before going into the applications with this short video, I would like to introduce some key concepts briefly. If you ask a different remote sensing scientist what vegetation is for them, you may have different responses because vegetation can be looked in many different ways depending on the research field and the final application of the remote sensing vegetation product. For example, vegetation is called fuel in the fire literature, but biodiversity in ecology. But whatever the definition is, as vegetation is the dominant component of most ecosystems, we can use remote sensing to routinely gather valuable information for characterizing and managing these organic systems. Why is this possible? Because the vegetation has a spectral signature very different from other land cover types. These spectral variations facilitate fairly precise detecting, identifying, and monitoring of vegetation on land surfaces, and in some instances, even within the oceans and other water bodies. Thus, we can continually assess changes in forests, grasslands, shrublands, crops, and so on, often at quantitative levels. The surface reflectance at two or more wavelengths can be combined into a single value to highlight a particular property of vegetation while minimizing the influence of external factors. This single value is called vegetation index. More than 150 vegetation indices have been published in a scientific literature and have been used, for example, to detect the presence and relative abundance of vegetation moisture, pigment, carbon uptake, etc. For example, the NDVI is the most well-known index used to detect live green canopies in multispectral remote sensing data. NDVI is calculated from the visible and near-infrared light reflected by vegetation. Healthy vegetation will have high values of NDVI since it absorbs most of the visible light that hits it and reflects a large portion of the near-infrared light. Unhealthy or sparse vegetation reflects more visible light and less near-infrared light, and therefore presents lower values of NDVI. Finally, bare soil presents very low values of NDVI, making it easy to apply a threshold to filter the soil influence out of vegetation applications. This animation shows monthly NDVI values from 2000 to the present, derived from MODIS. Dark green areas show where there was a lot of green leaf growth. Light greens show where there was some green leaf growth, and tan areas show little or not growth at all. This table presents a few other examples of vegetation indices grouped into two categories: greenness and water content indices. Greenness indices such as the NDVI, EVI, and the VARI combined reflectance values in the near-infrared and the visible region of the spectra to measure the general quantity and vigor of vegetation. Some applications include vegetation phenology studies, land use and climatological impact assessment, and vegetation productivity models, as we will see in the next videos. The EVI and the VARI differ from the NDVI because they include a band in the blue to minimize the influence of the atmosphere and also to be more sensitive to high vegetation cover. You may know that the NDVI tend to saturate in those situations. Water content vegetation indices use reflectance measurements in the near-infrared and shortwave infrared regions to take advantage of known absorption features of water. These indices provide a measure of the amount of water contained in the foliage canopy. Water content is an important quantity of vegetation because higher water content indicates healthier vegetation that is likely to grow faster and be more fire resistant. Consequently, application of these indices include canopy stress analysis, productivity prediction and modeling, fire risk analysis, and studies of ecosystem physiology. NDII uses a normalized difference formulation, and therefore the index values increase with increasing water content. The value of this index ranges from -1 to 1. The common range for green vegetation is 0.02 to 0.6. Finally, the moisture stress index is a simple ratio and therefore inverted relative to the NDII. Higher values indicate greater water stress and less water content. The common range for green vegetation is 0.4 to 2. Now that you know the basic of remote sensing and vegetation, you are ready to check out the next video in this series that will talk about forest land cover change mapping.